Entonces, bueno, pues buenos días a todos. Hoy tenemos el gusto de tener con nosotros a la doctora María Gutiérrez y ella es investigadora ahora de la Universidad de Harvard, está en el Brigham and Women Hospital y está en un laboratorio que se orienta a estudiar la autoinmunidad. Y la doctora Arcelus María es sobre todo una bioinformática, ella es es estudiante egresada de la UNAM, de la licenciatura en Ciencias Genómicas. Justamente estábamos hablando ayer con los estudiantes que ella es de la tercera generación y pues ha sido una generación, como lo verán ustedes, muy buena, muy prolífica en excelentes investigadores. Entonces, María hizo su licenciatura, como les dije, en la UNAM, hizo su doctorado en la Universidad de Ginebra y desde que terminó su doctorado se pasó a ocupar una plaza de postdoctorante en el laboratorio del doctor Sumya Rai Chanduri. Ustedes disculpen, exacto, pero no es un nombre muy fácil de pronunciar. Y en donde el interés principal de la investigación en ese laboratorio son estudiar las bases de la autoinmunidad, y para esto usan técnicas de genética, de bioinformática, de biología de sistemas y obviamente también de inmunología de wet lab, y conjuntan todo esto y para entender cuáles son los mecanismos moleculares que rigen estas enfermedades autoinmunes y explicar sobre todo un poco cuáles son las variantes génicas que se expresan y cómo es que se regulan esas variantes génicas en los diferentes individuos y qué tanto esto correlaciona con la susceptibilidad a desarrollar o no desarrollar enfermedades autoinmunes. Y esta creo que ha sido la línea de trabajo de María desde hace muchísimos años. Y bueno, sin más preámbulo, María, te agradezco muchísimo por estar con nosotros. Y pues, adelante.
Muchas gracias, Ivonne. Gracias por la invitación. Me da mucho gusto estar aquí. Me hubiera gustado estar en persona, sobre todo porque sería en mi hogar científico, pero bueno, estamos aquí virtualmente. Voy a compartir mi pantalla.
Perfecto. Entonces, quizás antes, en lo que haces esto, pues una vez más me gustaría pues agradecer a todas las personas que nos están acompañando por esta vía virtual, a las instituciones académicas que se asocian con nosotros semana con semana, y recordarles que pueden hacer preguntas a través del chat de YouTube y nosotros las leeremos aquí para que la doctora los vaya contestando al final de la plática.
Sí, perfecto. Por lo general, cuando estoy dando la plática no puedo ver las preguntas por chat, entonces...
Por eso las hacemos hasta el final y las leeremos probablemente para que las puedas ir contestando.
Muy bien. Me van a disculpar, pero voy a dar la plática en inglés. Ayer intenté un ratito dar mi presentación en español con los estudiantes y fue un intento catastrófico. Entonces, por el bien de sus oídos, la voy a dar en inglés. Okay. I'm going to start today with a very general background. I will then tell you about a first project where we studied cell state dependent dysregulation of autoimmune disease genes. And then finally about a second project where we discovered a transcriptional program that defines lymphocyte innate state. So I want to start with this oversimplified cartoon to illustrate that that our genome to a great extent dictates what genes are expressed and at which levels. And this to a great extent dictates the proteome of the cell, which determines the functions of a cell. So if you have a different transcriptional program, you have a different proteome and you have a cell with different functions. And then also you can apply perturbation to a cell, like if you stimulate a T cell, you can imagine how this perturbation creates a series of changes inside the cell, which changes the transcriptome and changes the cell state and what the cell does. Or you can have a genetic perturbation. This genetic perturbation can be like a coding variant. For example, it can alter the protein of a transcription factor, and you can imagine how this could alter substantially the transcriptome and alter the cell state. Or you can have a non-coding variant, and if this variant happens to alter the expression level of this gene, you can imagine that maybe this wouldn't have a great impact in the total cell state or transcriptome or cell state. But if you have a series of non-coding variants that each of them are affecting slightly the expression of a different gene, you can imagine how this could affect the cell state and affect the function of that cell. So with this, I want to just highlight that the transcriptome is very rich in information. And depending on what we measure, it's informing us about cell state, and this is informing us about immune function. And if we alter these things, this can lead to autoimmune disease. So within the transcriptome, there's different transcriptomic phenotypes that we can measure. There's gene expression, which is the thing that we mostly do. There's allele-specific expression. Each of us has 2 copies of a gene, one from our mother, one from our father. And when one of the 2 copies is expressed more than the other, this is called allele-specific expression. And usually this is driven by a genetic variant that is affecting the regulation of the gene. I apologize for the noise, that's very unexpected. Or we can have a score, a transcriptomic score that will summarize the information from hundreds of genes. Or we can measure alternative splicing and measure different isoforms that a gene is producing. And we can also make different comparisons. We can, depending on what transcriptomic phenotype we measure, we can, and what comparisons we make, we'll learn different things. So we can compare different cell types and learn about their commonalities and differences in function. We can compare different cell states and learn how the function of a cell type might change. We can compare single cells and try to understand heterogeneity within a cell type or understand transitional cell states. Or we can compare transcriptomes between different people and try to understand why some people end up developing disease and some not. So I'm going to start with the first project. These are all the people involved in this project. In particular, Yuri Baglenko did the experimental CRISPR functional assays. And many other people were involved. Jatin Arora performed key analyses for fine mapping. Susan Hannes did the time course experiments that I'll talk about. So for this project, I'd like to introduce the genetics of autoimmune disease. Genome-wide association studies have found hundreds of risk variants that are influencing whether you get an autoimmune disease. And these variants tend to be of small effects and non-coding. And people have found that these variants are enriched in regulatory elements that are specific for specific immune cell types. So, for example, for rheumatoid arthritis and type 1 diabetes, people have shown that these risk variants are enriched in regulatory elements specific for CD4 memory T cells. So the hypothesis is that these variants are affecting gene regulation, that together they affect T cell state, which then leads to intermediate immunophenotypes and then leads to autoimmune disease. And the better we get at understanding these mechanisms of how you go from a variant to dysregulation of genes and alteration of T cell states, the better we'll get at designing new therapies for these diseases. So this finding that a lot of these variants are are non-coding and probably affecting gene regulation has fueled a lot of studies that try to look for associations between genetic variants and gene expression, or also called EQTLs, expression quantitative trait loci. And here it's just a cartoon example. Each dot is a different individual, and you can see that individuals that have the GG genotype have lower gene expression levels, that individuals that have a GT genotype, which have lower gene expression levels than individuals that have a T genotype. And what was surprising for a lot of people is that when they were looking for these associations, it turns out that for most risk variants, we were not finding a co-localization with the EQTLs, or the variants that are affecting gene expression. A recent study estimated that only about 25% of autoimmune disease risk variants are co-localizing with these regulatory effects. And they looked at 3 immune cell types. So a big question is, where are the missing regulatory effects? So one possibility is that some of these effects are hidden in cell states that we haven't ascertained yet. And people have started to ascertain different cell states. For example, in T cells, these 2 studies stimulated T cells. And here's just an example of an EQTL or a regulatory effect that is active in cell state 2, but not in cell state 1. And if we zoom into a heterozygous individual here, this is a possible mechanism of how this variant could be driving a change in gene expression. So maybe this variant is affecting the binding of a transcription factor. So the G allele, the transcription factor, doesn't bind as well. So you have lower gene expression levels. And the T allele, the transcription factor, binds well. So you have higher expression levels of that copy of the gene. So this is, again, allele-specific expression. And maybe in this other cell state, maybe that transcription factor is not active or not expressed. So that's why you don't see that regulatory effect in that cell state. So back then, people had started to look at 1 or 2 stimulation states in T cells, but they had not looked at this in multiple cell states in like a high resolution time course experiment. So when I arrived to Shomo's lab, they had this pilot study where for 1 individual, they had isolated CD4 memory T cells, and they had stimulated them with anti-CD3, CD28 microbeads. And then they had performed RNA-seq at different time points after stimulation. And the original objective was to look for differential expression between these time points and see if there were some time points where there was a higher expression of genes in risk loci for autoimmune diseases. And coming from a functional population genomics lab, I knew that we could extract a little bit more information from this 1 individual. We could take advantage of heterozygous sites inside exons to look for allele-specific expression, which reflects the effects of genetic regulatory variation, and then look for dynamic allele-specific expression over time to look for cell state dependent regulatory effects. So just to depict how we are measuring allele-specific expression in a more graphic way, if you have a heterozygous site in an exon, well, you map your reads to the genome, and then you count for the reads overlapping this site, how many of the reads have 1 allele versus how many alleles have the other allele. And if this deviates from the expected 50-50 ratio, you call that allele-specific expression. And just to reiterate, when we're looking at this phenomenon, most of the time, we're actually looking at the effects of a genetic regulatory variant nearby. It can be this mechanism of the transcription factor, but it can also be other mechanisms like mRNA decay, alternative splicing, or others. So I want to show you first 1 example in the gene IL-10. IL-10, these are total gene expression levels. At 0 hours, you can see that IL-10 starts slowly expressed. And then when you stimulate the cells, it gets upregulated very strongly. It stays upregulated, and then after 24 hours, it gets downregulated. So if we look at one individual that has a heterozygous SNP in one of the exons of IL-10, and we quantify the reads from each allele, you can see that at the beginning, both alleles are more or less equally expressed. The alternative allele in blue, the reference allele in yellow. And then suddenly at 24 hours, you see a dominance by the alternative allele. And then this dominance stays at 48 and 72 hours. You can also look at this as the reference fraction that goes from 0 to 1. So the proportion of reads that come from the reference allele. And it starts more or less around 0.5. So both alleles express, and you can see the bias towards the alternative allele here. So what does this actually mean? This means that there will be some people that will be homozygous for the high expressor allele for IL-10. So they will be producing more IL-10 levels than individuals that are homozygous for the lowly expressed allele, right? And IL-10 is a cytokine with different functions, and you can imagine how producing different levels of IL-10 could alter, in theory, how the T cells of an individual mediate immune response. And IL-10 is within the risk locus for inflammatory bowel disease. And then, so this study is not yet showing that the risk variant in that locus is altering IL-10 regulation, but it's telling us that that now we know. I'm sorry, I'm going to interrupt 2 seconds. I'm going to tell them to interrupt the hammering. I don't know if you can hear it. Sorry, just 2 seconds. Okay, great. So that was one example. And the one thing that we found that was very striking for us in that first pilot individual was the most dramatic effects were coming from this gene called HLA-DQB1, which is the major risk gene for type 1 diabetes and celiac disease. And we will, well, I'll show you this example first. So this is a SNP in DQB1 for one individual. And at the beginning, you can see that it's the reference allele that is more highly expressed. And then suddenly at 48 hours and 72 hours, you have a strong dominance by the alternative allele. And so this is like a very strong allelic switch. And we were surprised by this because HLA-DQB1 is part of the HLA class 2 genes. And normally these are genes that code for antigen presenting proteins, and they're studied more in professional antigen presenting cells, not so much in T cells, but we do know that in human T cells, when they get activated, they express MHC class 2. And this does not happen in mouse. So that's partly why the function of these molecules in T cells has not been established yet. But we do know that in subsets that are T cell subsets that are expanded in rheumatoid arthritis patients, they tend to express MHC class 2. And the other thing we were worried about is that these genes are very, very polymorphic. So they are prone to have mapping biases at the bioinformatic level. So we wanted to make sure that this phenomenon we were observing was real and not just some bioinformatics artifact. So we developed a pipeline to quantify robustly allele-specific expression in HLA genes. And basically this is done creating an HLA personalized genome. So if you sequence or HLA type the HLA genes, you will know then for your individual allele 1 for DQB1 and allele 2. So you insert this sequence into your reference genome, as well as the sequences for the other HLA genes. And then you map your reads to the genome again, and you count the number of uniquely mapped reads to each cDNA allele. So now you're quantifying allele-specific expression instead of in a single SNP, you're quantifying it over these 4 exons, and they often include more than 20 mismatches between the 2 sequences. So here you can see again that the yellow allele is more expressed at the beginning, and then you have this switch of dominance towards the blue allele. So now we were sure that this phenomenon in this individual was real, it was actually happening. Then we ask, is this individual kind of weird, or is this a common phenomenon? So we repeated this pilot in another 2 individuals, and we saw again that the most dramatic effects were coming from this gene and other HLA genes. So we decided to go and do a larger scale study where we recruited 24 healthy individuals that were genome-wide genotyped. We sequenced their HLA genes. And we selected these individuals so that they would be heterozygous for DQB1 alleles. And we required more than 20 mismatches between the 2 alleles so that we could quantify robustly allele-specific expression for these people. And we repeated the experiment. So we isolated CD4 memory T cells. We stimulated them with anti-CD3, CD28 microbeads. And we performed RNA-seq at 8 different time points. And then we looked for dynamic allele-specific expression. So this is how we're detecting dynamic allele-specific expression. We're using a mixed effects logistic regression framework where we're essentially asking, is the log odds of observing the reference allele changing with time? So for each site, for each heterozygous site in each individual, you will have reads that have the reference allele. If you have a reference allele, you assign that a 1. If you have alternative allele, you assign that a 0. And then we're essentially measuring the effects of time and time squared. And we're including a random effect for sample to control for overdispersion. And for 2 individuals, we managed to get enough cells to create full time course replicates. And these replicates, they helped us define the best model to identify these dynamic allele-specific expression events. And the betas for time and time square are replicating well between these technical replicates. And the same if we take individuals that are 2 different individuals that are heterozygous for the same SNP, you can see how these effects are also replicating well. So in total, we found 561 events at 5% FDR. These events are spanning 186 genes. And I'm highlighting in this circle plot some of the examples of SNPs, like this SNP that is in CCR4, this SNP in UBASH3A. And we started noticing that a lot of these genes were in autoimmune disease loci. So we asked, are these events enriched in autoimmune disease loci? So the answer is yes. We found significant enrichment for dynamic allele-specific expression in loci that give you susceptibility to ankylosing spondylitis, celiac disease, vitiligo, type 1 diabetes, inflammatory bowel disease, rheumatoid arthritis, and multiple sclerosis. And then, and we didn't find enrichment in non-T cell mediated diseases like type 2 diabetes, schizophrenia, systemic lupus erythematosus genetically is more of a B cell driven disease, and coronary heart disease. And also if we compare this to whether we had looked at allele-specific expression at 0 hours only, or if we had performed a typical large-scale EQTL study in naive T cells, we we find more enrichment in disease loci for our dynamic ASC genes than if we had looked at 0 hours here in green, or at a public data set of naive T cell EQTLs. So for us, this indicates that probably a lot of these risk variants in autoimmune diseases are affecting gene regulation in a very cell state dependent manner. So we should look at multiple cell states to understand the mechanisms of these diseases. And then one other finding was what I mentioned already, this event in HLA genes. This is the MHC region is in chromosome 6, and you can see an accumulation of dynamic allele-specific expression events here. Most of these are falling in HLA genes. And I just want to illustrate some of the variability that we were seeing. So these are 3 individuals, and now I'm plotting here normalized allelic expression levels for DQB1. So you can see for this first individual, both alleles start low, and then the yellow allele stays low, but the blue allele spikes at 48 and 72 hours. For this middle individual, the 2 alleles start very differently, and then they converge at the very end to the same level. And in this third individual, you can see how the blue allele starts low and then gets upregulated at 48 and 72 hours, whereas the red allele is fluctuating more or less at the same level there. So we were surprised by this amount of variability, and then we start to ask, can we group these allelic profiles into discrete groups? So we found there's 3 main groups, or that we start calling allelic regulatory programs. So we have the late spike cis regulatory program, where you have that upregulation at 48 and 72 hours. Here on the heat map on the left, each row is a different allelic profile. And the color is the expression level. So you can see higher expression levels here for this group. We have the constant low regulatory program, and we have the fluctuating regulatory program. So you can be an individual that has 1 allele up here in the late spike program and maybe 1 down here in the constant low, or maybe you're an individual that has 2 alleles here in the fluctuating program, and that's how your alleles might be behaving when you stimulate your T cells. So our next question was, okay, these are kind of extreme effects at the RNA level. Are these effects translating to the protein level? And it's not so easy to quantify allele-specific expression at the protein level. So what we did is that we recruited 5 homozygous individuals for late spike alleles and 5 homozygous individuals for any of the 2 programs, and then try to measure protein levels with flow cytometry and see if there's differential expression between these 2 groups. So here I'm showing you an example of a late spike individual on the top and a constant low individual on the bottom. On the y-axis, we have CD25, an activation marker. And on the x-axis, we have HLA-DQ. And you can see that at the beginning, at day 0, both individuals have almost no cells that are DQ positive. And then as you stimulate the cells, at day 7, the late spike individual has 10% of its cells that are DQ positive, and the constant low individual has 3% of its cells of their cells that are DQ positive. So if we plot here the fold change between day 0 and day 7 for all the individuals, you can see how the late spike individuals have higher fold changes than the other individuals. So now it seems that, yes, this RNA dysregulation was translating to the protein level. Our next question was, okay, what is the genetic variant or genetic variants that are affecting the regulation of this gene? So long story short, we came up with these 6 candidate SNPs that were tracking with the late spike haplotype. So there are 5 SNPs here upstream of DQB1 and 1 SNP just downstream of DQB1. We performed ATAC-seq on a 72-hour simulated CD4 memory T cell sample. And you can see that the highest peak in the region is overlapping well this SNP that is the closest to the transcription start site. Other chromatin marks from public data were also supporting sort of active regulation here in this region. And we performed CRISPR experiments to see which of these SNPs might be affecting the expression of DQB1. And at the end, we found, well, we performed this CRISPR experiment in an in the HH cell line, which is a T cell line that expresses class 2. And we found that for this SNP, we isolated 8 clones that they still had the wild type. They were homozygous for the reference allele, and 8 clones that were homozygous for the alternative allele. And you can see that at both mRNA and protein levels, there's a higher expression for the clones that had that were homozygous for the alternative allele, which was the direction we expected. So to conclude, we found widespread inter-individual differences in dynamic gene regulation during T cell activation. We found that these events are enriched in autoimmune disease loci. We found pervasive and unexpected regulatory effects in HLA genes. And particularly for HLA-DQB1, we found 3 distinct transcriptional programs. And finally, we proved with CRISPR editing that a variant at the promoter is causing at least part of this T cell specific control of DQB1. I didn't have time to show you why we think this is T cell specific, but I am happy to tell you more about it in the questions. And then what are the implications of this study? So this study is highlighting that if we want to understand autoimmune disease mechanisms, we'll need to look at multiple cell states. This study is also highlighting that there's a large amount of regulatory variation for HLA genes, and we need to take more of these HLA personalized genome approaches to really document all the regulatory variation for these genes in other cell types and cell states too. It's also because we show that there's this very T cell and condition dependent regulation of class 2 in T cells. I feel like this is also highlighting that we should find the function of HLA class 2 in T cells. And also it's highlighting the possibility that in these HLA genes, it's very well known now that it's mainly the amino acid changes in these proteins that are driving your risk for autoimmune disease. But not all of the risk in that region has been explained by these amino acid changes. So there's still a possibility that also how much of this molecule you're expressing that can modulate your risk for autoimmune disease, and that remains to be determined. Okay, so I'm going to switch to the second project. This was a close collaboration between our group and Patrick Brennan's group. And these are all the people that were involved. So there are diverse subsets of human T cells. We have the conventional T cells that, well, the ones that have been more studied that recognize a wide variety of peptide antigens and express an alpha beta receptor. And they're usually, these antigens are presented by MHC class 1 or MHC class 2 molecules. But also there's this other class of less conventional T cells that are able to see other types of antigens presented by other types of molecules. So for example, iNKT cells can recognize lipid antigens presented by CD1D, MATE cells can recognize vitamin B derivatives presented by MR1. And there's also these T cells that express a gamma delta T cell receptor, and what antigens they are able to recognize and who presents them is not very well established yet. And what is particular about these cells is that they can get activated without the need of going through the T cell receptor, so they can get activated with cytokines, and they have sort of like they're known as poised effectors because they can have an effector response without a previous antigen exposure. So because of these properties, people often call them innate T cells, and they're involved in they have roles in allergy, in cancer, and autoimmunity. And back then they had been studied transcriptomically more in mice than in humans, and specifically in humans they had not been ascertained as a family. And so we were interested in characterizing transcriptomically these human innate T cells with the hypothesis that their shared effector capabilities may be driven by a common transcriptional program. Oops. So we immunophenotyped 101 individuals just to take a look at the frequency of these cells in peripheral blood. So you can see that mate cells, on average, they're composing 2.4% of T cells. NKT cells are less common, so on average they compose 0.09% of T cells. V delta 1 cells is 1.25%, and V delta 2 cells are 4.7%. And it's very variable. In total, they compose 1 to 25% of peripheral blood T cells. So we used PBMCs from 6 healthy donors. From these, we flow sorted these 7 cell types. So these 4 innate T cell subsets that I talked about, V delta 1, V delta 2, mate, and iNKT cells. We wanted to compare them with the conventional adaptive T cells, so CD4 T cells and CD8 T cells. And then we also wanted to compare them with an innate comparator, so we chose natural killer cells. And we performed low input RNA-seq on 1,000 cells in duplicate. So this is our gating strategy. We used tetramers to isolate mate cells, NKT cells, and the V delta subsets. So first, what we did is we quantified gene expression levels, and to reduce the high dimensionality of our data, we performed principal component analysis to capture the major axis of variation. So you can see that on PC1 here, cells are clustering. On one side, you have the CD4 and CD8 T cells, and on the other extreme, you have the NK cells, which were our innate comparator. And in the middle, you have the innate T cells. So if we plot here the PC1 score for the different cell types, you can see how they are forming a gradient from most adaptive to most innate. And because of this like continuum or gradient, we started calling the innateness gradient, because they go from most adaptive to most innate. And we really wanted to know, okay, what is behind this gradient? What are the genes participating in this gradient? So we used a linear mixed model to find genes that were associated with this rank order of the gradient that was given to us by the PCA. So here's an example of an innateness gene, because it's associated positively with innateness. And then this is an example of an adaptiveness gene, because it's associated positively with adaptiveness or negatively with innateness. So we found, and here we were controlling for donor as a random effect. And our gradient, as I said, it's a rank order of 1, 2, 3, 4, 5, 6, 7. So we found 1,800 genes associated with this innateness gradient. about 1,000 of them positively associated with innateness, depicted some examples here. Each row is a gene and the color is the expression level. So you can see these genes are more highly expressed on this side. There were 797 genes that were more adaptiveness genes, so they're more highly expressed on this side. And there were both protein coding genes and link RNA genes that were behaving like this. So what is behind the innateness genes? Who are these genes? What pathways do they belong to? So we found that a lot of these genes had roles in cytotoxicity, migration, and cytokine and chemokine genes. So here's an example of our volcano plot, where x-axis is innateless level, or the beta in our model, and y-axis is the minus log 10 p-value. And I'm coloring in blue all of the genes that have these Go terms, NK-mediated immunity and cellular defense response, that was enriched for innateness. So on this side of the volcano plot, and I'm highlighting here some example genes like granzyme B, TYRO-BP, granulysin, perforin, CXC3R1, and some key receptors. And also, a lot of the cytokines and chemokines are not very highly expressed. But if we sum the total transcription levels of these cytokine and chemokine genes that were expressed in our data set, you can see how the total transcriptomic mass of these cytokines and chemokines is also following this innateness gradient. And I don't have time to show you, but we performed some experiments where we showed that that the mRNA of interferon gamma is actually in the form of preformed mRNA. So the RNA is there ready to be immunity translated when you stimulate cells. So we think this is contributing to some of the properties of these cells of early cytokine production. And also we observed that there's more active translation in this innate T cells compared to the adaptive T cells. So this is just to show you, these are known things in the field, that these cells, they have early cytokine production and they can get stimulated independently from the TCR, so just with cytokines. So here on the left, you can see how innate T cells are, on average, have high proportion of interferon gamma positive cells after 5 hours of PMAI stimulation. And also, if you stimulate them with IL-12 and IL-18, or IL-12, IL-18 and interferon alpha, these innate T cells, there's many of them are producing interferon gamma, whereas for the CD4 and CD8 cells, it's just a very tiny proportion that do. And what's in the other side of the gradient? What's in the adaptiveness genes? So when we did a pathway enrichment analysis, we were finding a lot of terms associated with translational machinery. So our top term was cytosolic ribosome, and here's an example of a ribosomal protein gene that you can see very clearly its adaptiveness association. So it's higher in CD4 and CD8 cells. We then realized that it was not only the ribosomal protein genes, it was also elongation factors, such as translation elongation factors, such as this one. And then our top transcription factor that was associated with adaptiveness was MYC. And MYC is known to regulate ribosomal RNA. So we also did a qPCR to look for a ribosomal RNA precursor, and we saw how this was also associated with adaptiveness. So we were thinking, what does this actually mean in terms of the function of these cells? And we thought, well, maybe having higher regulation here, higher transcript levels overall of all this translation machinery can give you a higher proliferative capacity. So we performed an experiment where we stimulated all of our T cells. We couldn't stimulate NK cells because it doesn't have a T cell receptor, and we stimulated them with anti-CD3 and CD28 beads. And you can see clearly that the division index at day 3 is higher for CD4 and CD8 cells compared to the innate T cells. So this is like a middle conclusion slide where, just as a recap, we found this innateness gradient, or it's a transcriptional program that it's like gradually turn on or off, and it determines your innateness state. So the more you are turning on this transcriptional program, the more you're upregulating the effector transcriptome, and we think this is leading to that property of early cytokine production in these cells. And also the more you're turning on this program, you're actually downregulating ribosomal machinery transcripts. And we think that this is causing these cells to sacrifice their proliferative capacity. So we were also wondering whether there was heterogeneity within these subsets, right? So we did a single cell RNA-seq experiment with cell hashing. This is the Stokios et al. Protocol, also known as CiteSeq. So what we did is we sorted our cells like we did in the other experiment, and each cell gets labeled with a general antibody that has a specific barcode. So then you pool your cells, you run them through the 10x machine, you build libraries for mRNA and for these hashtag oligos. And after that, you can know each cell, which cell type it belonged to. So here I'm showing you the cells in UMAP space based on the top 20 PCs. And you can see again that the CD4 and CD8 cells, most of them are clustering up here. Then you have this intermediate group with mostly MATE and iNKT cells. Then the gamma deltas, you have BD2 and BD1 here. And then at the corner here, you have natural killer cells. So if we took the data from our low input RNA-seq experiment and created an innateness score based on that PCA. And so this innateness score is telling us how much of that innateness program you have turned on. And if we color the cells by that, you can see that it replicates the more adaptive cells have lower innateness score, and then the more, and you see the gradient, and the more innate cells have the highest innateness score. So we performed some clustering, and we found these four main groups that we start calling the four innateness states. And what is interesting is that for a given cell type, you have heterogeneity. So for example, for CD8 cells, you have most cells in the group 1, but you also have cells in group 2 and group 3. So we wanted to, we also observed this heterogeneity for other of our subsets, but we wanted to confirm this in other settings. So we used public data sets. So in this data set, they used microarray data where they isolated HCMB specific CD8 cells from infected individuals. So you can see that naive CD8 T cells have lower innateness score than memory CD8 T cells, which have lower innateness score than the effector CD8 T cells. And this is also true for CD4 T cell subsets. So in this transcriptomic data set from the lab, you can also see, again, naive cells have lower innateness score than effector subset. And what was interesting is that the subset that was shown to be expanded in rheumatoid arthritis patients was the one with the highest innateness score. And we also used a single cell data set from breast cancer patients. And you can see how the T cells from the tumor have higher innateness scores than the T cells from healthy breast tissue. So in conclusion, besides this transcriptional program being a balance between the effector transcriptome and early cytokine production and sacrificing proliferative capacity, we also observed that this innateness state varies within CD4 and CD8 T cell subpopulations, and it's increased in an autoimmune pathogenic subset and the tumor microenvironment. So with that, I would like to thank all the people involved in these projects, in particular, Shomo Ray-Chowdhury, my advisor, who has been a great mentor, and also my collaborator, Patrick Brennan. And I would also like to tell you about my future directions. I'm starting my own lab at Boston Children's Hospital early next year. It's going to be a regulatory genomics and systems immunology lab, mainly focused on pediatric onset immune-mediated diseases. And I'm starting to look for people, especially if you're interested in B cells and lupus. Shoot me an email. And I also have a collaborator in Boston Children's Hospital that is looking for a neutrophil postdoc. So thank you for your attention, and I'll take questions now.
I'll read it to you, Maria.
Yeah, I'm reading it now. So
okay,
okay. I can
maybe
read
you
it
can read it so that other people know what the question is.
Yeah. So Diego Arturo is asking, you showed us that this quick and robust poise effector by the innate T cells is driven by a transcriptional program, a pool of mRNAs that wait to be translated. Other studies have demonstrated that epigenetic mechanisms can prime these genes in other immune cells by using link RNAs. Are you aimed to describe this mechanism in innate T cells? That's a great question. We did find interesting things with link RNAs. Particularly, there was one that was near the ID2 transcription factor that was being co-expressed with ID2. So we thought it probably had a role in regulating ID2, but we weren't able to follow up this in very detail. There were too many sort of things to follow up on. So unfortunately, we haven't follow up on that, but it's a very interesting question that we could do in the future. Okay, another question by Kevin Galindo. Are there any differences between innate and MHC restricted T cells that could lead to the discovery of mechanisms for avoiding autoimmune disease? Also great question. Are there differences between innate and yeah, maybe. I think innate T cells are more understudied, so definitely we need to study them more closely, ascertain their epigenome, their transcriptome, and first assess, at least from my genetic point of view, whether some of these genetic susceptibility to autoimmune diseases are being mediated through these innate T cells. And if so, how are they doing that? And then eventually, how, if we could modulate these innate T cells, could we prevent or cure disease? Yes, but lots of work to do in that area. Okay, Itzel Fernanda Pineda, studies with longer periods of time, like different life stages, could yield something meaningful about the allele-specific expression? Yes, definitely. Since here we're showing a lot of gene by environment interactions, right? Depending on the environmental context of the cell state, you observe that regulatory effect at the allele-specific expression level. So I could imagine that also age will change your cell state and also alter these regulatory effects. So that's a great point that maybe we should also take into account for how these diseases are developed.
That's right.
Emmanuel Castañeda Castro, I guess that the expression of the alleles have to be so regulated. Did you know some mechanism in common of regulation upstream that could avoid or decrease the effect of these risk alleles? A mechanism in common of regulation upstream. Yeah, that's a good point. I'm trying to think of it. I guess, yes, if there's if you know that this transcription factor driving this regulation, you can you can try to modulate the activity of that transcription factor if it's affecting many genes that are involved in this autoimmune pathogenesis, although you should make sure that you're not altering other things too, right? But yeah, it's an interesting question and something we should think more about. Rodrigo Cervantes, do you know if B cells could deploy the adaptiveness innateness gradient? Not the very same one, but something similar perhaps? Yeah, that's a really good question. Yeah, we did have, for one individual, we had B cells in this project, and I'm trying to remember where innateness gradient they were falling. I think they were kind of clustering separately. So, but definitely maybe within B cell subsets, we could try to see if there's a parallel transcriptional program. Yeah, that would be great to see. And I imagine this transcriptional program seems very common, and now we have other T cell data sets in the lab, and we keep seeing this innateness gradient. So yeah, I wouldn't be surprised if at least part of this program is also present in B cells. Medina Ale, hola Ale. Maybe I missed it, but was there any sex effect that you could detect? I'm guessing you're asking on the allele-specific expression part. We haven't looked for sex effects in this data set. People that look for sex effects in these regulatory effects, like EQTL studies, it's kind of tricky to find them. It's not so easy to find them. So I think we would need a very large sample size to detect that. But we could definitely try if within our 24 individuals, maybe there's a strong enough effect that we can see. Definitely worth a try. Thank you.
I guess that will be it then. I had a question too. And regarding the dynamic allele-specific expression that you found in T cells through MHC class 2, is this also present in other cells, like in B cells or antigen-presenting cells?
Yes, I will. I don't know yet. There's another dynamic EQTL paper in, it was cells that are differentiating into cardiomyocytes. So I do expect to see this kind of effects in all cell types.
The same individual.
Yeah, so I would like to explore this in the future, especially in B cells in the context of lupus.
Right.
Yeah.
That seems like a good point to start.
Yeah.
You already have part of the story.
Yeah, we already have the methods.
Well, that's not too bad.
The computational ones. Yeah.
Okay, are there any other questions?
I think maybe one. Yeah, Victor Eduardo Nieto Caballero. Since there are different SNPs in one locus that may drive susceptibility for different diseases, or that the same allele can increase risk for one disease but be protective for another, what do you think are the main mechanisms behind that? Yes, that's a great question. Yeah, how can it, especially, yeah, I think when you have the first scenario where you have different SNPs in one locus that may drive susceptibility for different diseases, yeah, maybe they're slightly affecting differently regulation of this gene, and then maybe together with combination of other risk alleles, it ends up dysregulating some pathway that is important more for one disease than the other. Maybe it's not. I mean, I don't think anyone knows this. And also, and then for the one allele that is protected for one and a risk allele for the other one, that's yeah. I don't think people know this yet, but it's a great question. Yeah, I don't know how to answer it, but it's a really good question. We should find out.
So there is someone, Fernanda has a question.
Fernanda Garcia. The difference in concentrations of cells in an innateness gradient between people of different ages has specific consequences on the immune response. Yeah, that's a really good question. So we did find an association in this immunophenotyping of these 101 individuals. We did find associations with age for, I think it was mainly 2 subsets. One of them was mate cells. I don't remember the other one, but there was, as you became older, you had less mate cells. I think it's partly because we have this relative quantification. So maybe the older you get, the more exposed you get to antigens. So maybe the more expansion you have from CD4 and CD8 T cells. And maybe that is driving that effect. But it could also be that maybe your mate cells get reduced. We cannot disentangle those 2 scenarios with our data. But yeah, I imagine that this would have consequences on the immune response, but I don't know what they are.
So I have another question that came into my WhatsApp. So I'm going to read it to you, okay?
Okay.
So it's from Gustavo Pedraza, and it's in Spanish. Felicidades, muy interesante trabajo. Mis preguntas son, 1, ¿el alelo con la variante GG que genera mayores niveles de HLA en linfocitos activados está asociado con mayor susceptibilidad a desarrollar la enfermedad? Y si es así, ¿qué mecanismos proponen y qué factor transcripcional regula la expresión diferencial? ¿La activación induce la expresión de un factor de transcripción positivo o reduce la expresión de un regulador negativo?
Sí, muy buenas preguntas. La voy a responder en español. Sí,
sí.
Bueno, voy a intentar, y perdón si meto muchas palabras en inglés. El SNP que vimos en el promotor, intentamos ver en diabetes tipo 1, porque este es el gen de mayor riesgo para diabetes tipo 1. Entonces, vimos en un estudio de GWAS y sí está asociado a la enfermedad. Particularmente, si se acuerdan del haplotipo que es el H-spike, que tiene la regulación más alta a 48-72 horas. En ese haplotipo había muchos alelos de DQB1 que más bien son protectivos para la enfermedad de diabetes tipo 1. Pero no podemos desenredar los efectos de la variante en el promotor con los polimorfismos de aminoácidos. Entonces, tenemos que ver en un estudio que sea con un tamaño de muestra muy, muy grande y en un estudio de esos ya vamos a poder ver, ok, los que tienen la susceptibilidad por el aminoácido, dentro de ellos hay algunos que tengan el alelo 1 del promotor versus el alelo 2 y hay ahí una asociación con mayor riesgo, ¿no? Ese sería el siguiente paso a hacer para ver si ese nivel de expresión sí está influyendo en la enfermedad. Y en cuanto a qué factores regulan, no logramos saber. El SNP no está afectando directamente ningún seed motif de ningún transcription factor. Sí hay datos de ChIP-seq, de ENCODE y de otros de factores de transcripción que se están pegando ahí en esa región, pero no sabemos exactamente cuál está modificando. Y con un experimento de EMSA que hizo Yuri, sí vimos que hay un complejo nuclear que se pega de manera alelo-específica en la HA cell line. Y por cómo se está pegando, puede que pensamos que tal vez es un complejo represivo y que está reprimiendo DQB1, y si tienes la variante alternativa, no se está pegando tan bien y tal vez eso te permite esa sobreregulación.
Claro. Bueno, dice que muchas gracias.
Nada.
Bueno, pues yo creo que si ya no hay más preguntas, pues María, no nos queda más que agradecerte. ¿Tú ves
Creo
más
que hay una
preguntas?
más, no sé si...
Una de Daniela Fernández, sí.
Daniela Fernández. Talking about the individual innateness metric, Do you think that a higher innateness would be reflected as an increased or more aggressive response to infections? Sí, muy buena pregunta. Pues esa sería mi intuición de que sí, pero sí, pero no sabemos. Y definitivamente sí hay algo, un poco de paralelismo entre este programa de innateness y cuando activas unas células. Sí hay como un common access o variation ahí, pero tenemos que explorar mejor cuando active las células, el activation programs y qué tan parecido es al innateness program.
So we have a lot of thank you.
Pues muchas gracias, gracias por sus preguntas y gracias
Sí,
por invitarme.
Pues muchas gracias y muchas gracias por estar con nosotros y realmente fue una plática muy interesante y pues mucha suerte en lo que viene ahora.
Gracias, gracias Ivonne. Nos estamos viendo en conferencias de inmunología.
Órale. Y bueno, a lo mejor por ahí luego te preguntamos cosas también.
Sí, lo que quieran.
Muy bien. Pues muchísimas gracias a todos y bueno, pues nos vemos entonces la semana entrante. No sé si es a la misma hora, pero bueno, más o menos. Nos vemos, María, muchas
Bye.
gracias.